Hello everyone, my name is Sayeh Kazem and today I'm going to present my research project which tries to map the effects of genomic deletions and duplications on cognitive ability. I will start with a bit of background. Copy number variants or CNVs are DNA segments, deletions or duplications with potential consequences on cognitive ability. Cognitive ability is known for its high prevalence rate of mental disorders among people with intellectual disability. Moreover, its heritability is estimated at around 50 to 80%. Research studies show that haploinsufficient genes measured by love modulate the effect size and cognition when deleted or duplicated. Here you can see the overview of biological interaction between genes, tissues, cells, GO-terms, cognition, and neurodevelopmental disorder. In this project, we hypothesize that the functional information of genes can mediate the observed association between CNVs and cognitive ability. And the aim of this project is to understand and predict the effects of CNVs on cognitive ability using this information, in addition to loss of function information. Here I illustrated that our pipeline in which at left you can see the A panel that represented our data set, which shows the expression level across cell types, tissues, jig tests, and also incorporating the information of GO-terms. The first thing that we did, we computed the distance in each score group and then to extract the weighted average of distances. This weighted average of distances is considered as an input of the FATE algorithm, which FATE algorithm is a kind of a new dimension reduction methodology which worked properly for biological data. So we extracted two kinds of information from this FATE algorithm. The first one is to extract the informational distance. So for that, with that, we're gonna apply neighbors analysis by searchlight algorithm to estimate the gene dosed effect sizes on cognition. So in that case, the idea is to compute for each specific gene, we're gonna consider the number of genes inside the neighbors, which you can see by dark red color, and the number of genes outside that neighbor, which color coded by black. So then we're gonna apply the regression model, in which in the regression model the cognitive ability is considered as an output variable, and two input variables are the number of genes inside the neighbor and the number of genes outside neighbor. In this analysis, our main and interested effect sizes is beta 1, which is the effect sizes which is the estimation of for the variable that counts the number of genes inside the neighbor. The other information that we're gonna extract from this algorithm is actually dimension reduction using MDS for visualization in 2D. And then here at bottom you can see the summary of the data set in which we have in total around 258,000 healthy individuals. Now I'm going to show some preliminary results that we got. At A panel you can see we illustrate all the genes in by FATE manifold. Then we extracted the genes with deleted genes and duplicated genes separately in our data set, and then just visualize the genes that have the significant effect sizes. I should mention that we also divided this in two panels, B1 and B2, in which B1 represent the most intolerant genes, the LOF score less than 0.35, and B2 for the tolerant genes with a LOF score more than 1. And the same thing, we did the same thing for duplication. And as a in general, by comparing these manifolds and gradients, you can see that there is a higher negative effect sizes for the gene with deletion, both intolerant and tolerant genes, in comparison with the genes with that are duplicated. So to check if these differences are significant or not, we did a t-test and chi-square test. And what we found is that deletion decrease more cognitive ability than duplications, which we did the test for that, and showed that this is significantly different. The direction of the effect sizes on cognition are highly dependent on how much genes are mutation intolerant. We also observed that 90% and 85% of intolerant genes decrease the cognitive ability when deleted and duplicated, respectively. 33% of tolerant genes increase cognitive ability when subjected to either deletion or duplication. And all in all, we found that these findings emphasize the complex relationship between genetic mutations and cognition performance. And we are still processing this analysis. Thank you.